merismatic tissues or meristems are tissues that have the ability to stretch, enlarge, and differentiate into other new types of cells. These tissues possess the ability of continuous division and are responsible for the new formation of plant cells.
Meristems are actively dividing tissues of the plant. They are responsible for the primary elongation and secondary thickness growth of the plant. All new organs and their growth occur at the division of the meristematic tissue. Secondary tissues such as wood, cork, are also formed due to the activity of the meristematic tissue.
The classification of meristematic tissue. Based on origin, from meristem or primordial meristem, in bionic meristem, it originates to the primary stage of meristematic tissue. This represents two plant and bionic axes to this type of meristem, and it gives rise to the primary meristem. The primary meristem, it originates from the promeristem and can be found at different planes of the plant, for example, the root or shoot of the plant. The secondary meristem, it originates from primary meristem and can be found at the cambium of the plant, and it gives rise to the secondary permanent tissue during the secondary growth.
Classification of meristem based on position. 1. Apical meristem. It is present in shoot and root apex. It also divides in different planes and helps in increase in length. Second is intercalary meristem, found intercalated between permanent tissue or base of internode. This also helps in elongation of plant body. Third, lateral meristem. This presents in lateral side of the plant body, and it also divides periclinal and it also helps in increase the thickness or girth during secondary growth. Example like vascular cambium, cork cambium, interfascicular cambium. Mass meristem. It divides in all planes and form a mass of cells. For example, like endosperm. Rib meristem or file meristem. It divides in a single plane and form a row of cells. For example, like cortex and pith. And the next is plate meristem. It divides in two planes and form a plate of cells, like epidermis and epiblema.
Based on function, protoderm, it develops epidermis tissue system. Ground meristem, it develops cortex and pith. Procambium, it makes primary vascular bundle.
Histogen theory. Histogen theory was proposed by Hunstein 1868. This theory is applicable to both shoot and root apex. In this theory, there are three zones differentiated in apical meristem. These are the outer dermatogen, middle perioblem, and innermost perome. Outer dermatogen gives rise to epidermis or epiblema, which is found in the roots. Middle perioblem gives rise to cortex, while the innermost perome forms steel or vascular tissue.
Apical cell is the structural and functional unit of the meristem. The apical cell theory was proposed by Hofmeister and was later then supported by Carl Nageli in 1858. According to this theory, a single apical cell is responsible for the development of all aerial plant parts. This theory is applicable in algae, bryophytes, and pteridophytes, and not in the phanerogramms. The formation of shoot apex from a single superficial cell that is known as apical cell.
Tunica purpose theory is proposed by Schmidt during 1924. This theory is to explain the shoot apical meristem of the plant. According to this theory, the shoot apical meristem is a cone-shaped structure that is protected by the leaf primordia. Like the San Francisco plant, you can see here the shoot apex or the shoot apical meristem. The shoot apical meristem has two main regions, the inner corpus and the outer tunica. The tunica is the outer zone that consists of one or more peripheral layers. In the cotyledons, it consists of 1 to 5 layers of tunica cells, while in monocotyledons, it consists of 1 to 3 tunica cells. The tunica cells are much smaller than the corpus cells and they undergo anticlinal divisions. The outer layer of the tunica cell forms the epidermis, while the other layer forms the leaf primordia or the hypodermal cells. Inner corpus are the central undifferentiated multi-layered mass of cells. These cells are surrounded by the tunica cells and are much comparatively larger. These cells are divided in irregular divisions and form procambium and ground meristem.